Hello everyone, I'm Xiaoyu from Tianjin University. It's my honor to be here to show you my work about Plasmonic Micropillar-based SERS sensing platform for integrin detection of regulated cells. So let's begin with the first question. What is integrin? Integrin is a family of transmembrane protein. It serves as the linker between cell cytoskeleton and extracellular matrix. It can mediate the cell response to the artificial ECM of signals such as force, rigidity, and ligand distribution. It plays an important role in cell adhesion, endocytosis, and cytoskeletal organization. So modifying the variation and the distribution of integrin when cells are cultured on the artificial ECM is of vital importance to rival the mechanisms of cell ECM interactions. When cells are cultured on the artificial ECM, the common method to characterize integrin is immunofluorescence, just like this figure. It shows the distribution and the amount of integrin, but this method is limited to fixed cells. To detect integrin of living cells in a real-time, low destructive and sensitive method, surface-enhanced Raman scattering has attracted greater attention. It can show the component information by molecular fingerprint signals. But to enhance the signal intensity, plasmonic materials is needed, which do not exist in the majority of artificial ECM. So in our work, we propose a plasmonic micropillar by introducing gold nanorod on the surface of PDMS micropillar. We use this structure as the artificial ECM. To investigate the cell-ECM interactions, cells were cultured on the plasmonic micropillar by the topography of PDMS micropillar. And at the same time, the gold nanorods assembly on the PDMS micropillar can serve as the cell substrates for integrin detection. This slide shows the fabrication process of the PDMS micropillar. By spring coating photoresist, lithography, deep silicon etching, and removing the photoresist, silicon home master was achieved. After that, the PDMS micropillar structure was achieved by replica molding on the silicon home master. This image shows the morphology of silicon home master and the PDMS micropillar. It verifies the conformity and the uniformity of the PDMS micropillar. The outline of the PDMS micropillar shows that each pillar is 4.4 micropillar in diameter, 3.5 micrometer in space, and 5.9 micrometer in height. After that, three methods were proposed to introducing gold nanorod on the PDMS micropillar to achieve the plasmonic micropillar. The first method is solvent evaporation. That means simply dropping gold nanorod solution on the surface of PDMS micropillar and let the solvent evaporate. The gold nanorod assembly was formed when all solvent has evaporated. The second method is absorption transferring. By mixing PFT with gold nanorod solution, a dense film of gold nanorods was formed on the water-hexane interface. After the gold nanorod film floated up about the hexane, a piece of PDMS micropillar was placed near the gold nanorod assembly, and the plasmonic micropillar was formed by absorption transferring. The third method is very similar to the second one, but it replaced the PDMS micropillar with a flat one. The gold nanorods modified flat PDMS structure was contacted with the PDMS micropillar, and the plasmonic micropillar was achieved by stamp transferring. This figure shows the morphology and the dimension of the gold nanorod. After the three methods, the final forming plasmonic micropillar was shown as the SEM images and the confocal reflection images, where the gold nanorods reflect red glow. As shown in this slide, you can see the gold nanorod assembly on the PDMS micropillar is not uniform by solvent evaporation-driven assembly, where scale-up homogeneous assembly was achieved by absorption transferring and stamp transferring. The differences between the two methods is that gold nanorods fall into the gaps between the PDMS micropillar, or they modified only on the top of the PDMS micropillar. to estimate the SERS enhancement performance and homogeneity of the plasmonic micropillar. The SERS spectra of form BA were collecting at different testing points. As shown in this slide, the solvent evaporation shows a high relative standard deviation, where the stem transferrin has the lowest relative standard deviation, that is 7.3%, and a higher intensity than the counterpart of the method of absorption transferrin. So the third method was chosen for subsequent cell experiments. To investigate cell ECM interactions, the cells were cultured on the plasmonic micropillar, and the gold nanorods modified flat PDMS was used as a flat control. As shown in this figure, the cells grew along the plasmonic micropillar, grew along the x-axis of the topography cues, where the cells in the flat control show no specific directions. The alignment distribution along 0 degree and 90 degree increases when cells cultured on the ECM with micropillar topography. It verifies the cell regulation was achieved by plasmonic micropillar. After that, the integrin distribution was imaged by immunofluorescence and a confocal laser microscope. As shown in this slide, when cells are cultured on the plasmatic micropillar, the nucleus becomes longer and thinner, and more integrin is distributed near the micropillar, and the distribution of integrin and the cytoskeleton is highly consistent. Well, when cells cultured in the flat control, the cells grow along all directions, and the nucleus is more circular, more integrin distributed near the nucleus. Although integrin can be imaged by immunofluorescence, detect integrin distribution of living cells is still lack. So we try to measure the SERS spectrum of integrin, but the SERS spectrum of integrin is really weak and drowned out by the interference. So we introduce MCR analysis to solve this problem, which can estimate the pure component spectral and the contributions of each component from complex medium. The two Raman tags with different characteristic peak, that is Feng B and Feng PY, were used to test the performance of MCR analysis. The red line shows the spectrum of Feng B, and the black one shows that of Feng PY. And these are the mixture solutions of the two Raman tags with different proportions. The traditional component discrimination is using peak intensity ratio, just like figure B and figure C, but it suffers great error from spectral overlap. However, MCR analysis uses the whole spectrum instead of the single characteristic peak, so it improves the performance of component discrimination. The third spectrum of the plasmonic micropillar with or without integrin are shown in this figure. And MCR analysis was used to get the pure component spectral and the contributions of each component. The pure component spectral resembles strongly with the marrying results, and the contributions of integrin spirit with that of the gold nanorods. It verifies the detection of integrin was achieved by SERS detection and MCR analysis. The contributions of integrin is more than the counterpart on the flat control. The cells, when cells are cultured on different ECM, the distribution of integrin may be shown like this figure. That the cells distribute more integrin at the binding positions for capturing the micropillar, while distribution and the less distribution on the flat control. Further experiments is ongoing for investigate the cell ECM interactions. In conclusion, we propose a plasmonic micropillar, which can serve as not only the topography cues for cell regulation, but also to serve substrates for integrin detection. The cells cultured on the plasmonic micropillar grow along the topography cues and show more integrin. Okay, that is my report, and many thanks to the National Natural Science Foundation of China, many thanks to Tianjin University and my research group. And many thanks for your listening. Are there any questions?